Let us discuss the types of animal cell culture and cell lines. There are two types of cell culture, that is primary and secondary cell culture, and it's broadly classified, as well as cell lines. That is primary cell culture, in which the tissues are derived from the host directly. The parental tissue normally adjacent cells are connected by connective elements. That connective elements are in turn degraded. What are all the connective elements which are found in the system? That normally it's trypsin or collagenase is the enzyme that are used to degrade or disaggregate individual cells from the fragment of tissue. Then these individual cells are further added to the culture medium for the establishment of the primary culture. This addition of trypsin as well as collagenase is responsible for the breakdown of the connective elements like connexin, adenosine, all those things are in turn and degraded by this mechanism, this enzymatic mechanism. Then further these cells become individual cells. These cells are further used for adherent culture as well as for the suspension culture. The disaggregated individual cells from the primary tissue are then taken up and subjected to adherent culture. What is the difference between adherent culture and the suspension culture means? In the adherent culture, a substratum, that is the bottom of the culture vessels, are responsible for the growth and development. So it is going and attaching to the substratum and it develops as a monolayer, just like a film. It is growing. Adjacent cells are connected and it is growing as a film in a semi-solid surface in the case of the adherent cell culture. And it is immobile, and moreover due to the network of the connective tissues which are present in it. Normally the fibroblast and epithelial cells are growing like a network of connective tissue. At the bottom of the cell culture, these fibroblast and epithelial cells are forming a continuous layer of cell with one cell in thickness. That is, only one cell thickness will be there as a monolayer, we call it as. When it grows, only one adjacent cells are there individually, just like placing a coin one by one adjacent to each other as a single layer, we call it as a monolayer. So same manner these cells will grow by attaching to the bottom of the culture vessel, and we require some scraping elements are needed for the removal of the cells from these culture vessel for this adherent. Adherent means sticking to the surface. So these are scrapers are utilized for the removal of the cells under this process. So when we view these under inverted microscope, we can see these cells are growing as a single layer in the case of this. Whereas in suspension culture, it doesn't attaches to the substratum or the bottom of the culture vessel, just it is present in a culture medium which is liquid in nature. It is not semi-solid. So it is not attaching to the surface, then we are call it as anchorage independent or non-adherent cells. Especially the hematopoietic stem cells derived from blood, spleen and bone marrow and tumor cells are example of suspension culture. These cells grow faster when compared to the adherent culture because the cells are free in the medium that receives the nutrients in sufficient amount as well as the oxygen for its growth and development. It is homogeneous development takes place when compared to the layered culture. Once this is grown, we take up these primary cell culture and we subject them to subculture or passage. That is, the resulting culture is called as secondary cell culture. During the subculturing culturing, a portion of the cells are transferred to the new vessel having a fresh medium for the continuous growth of the cells. Normally, the subculturing of the primary cells leads to the generation of secondary cell lines. So we come across finite cell lines, continuous cell lines, we get it. There are two types where the cell lines are categorized. What is finite cell line means? The cell lines are limited number of cells are undergoing cell division and have a limited span of time called finite cell lines. That is non-proliferative. After a period of time, it cannot able to proliferate. It reaches the Sassanen stage or G0 stage in the case of the cells. We call it as finite cell lines. These cell lines are derived from the primary cell culture which are utilized. During the passage within the cells with highest growth rate capacity which predominates to uniformity in the phenotype as well as in the genotype of these cells during the cell culture, that is continuous cells in the continuous cell line. Some cells of undergoing finite cell line undergo transformation and has the ability to divide indefinitely to form the continuous cell lines. This type of transformation or mutation can occur spontaneously or by the addition of some chemicals or by viral induction. These cell cultures are prepared and by this way and subcultured to form the infinite permanent cell lines or immortal cell lines, or we call it as tumorogenic cell lines. These especially utilized for the purpose of cancerous cell line testing and alterations. It is less demanding for nutrients when compared to the previous culture. How to maintain the aseptic condition means normally the cell cultures are subjected to bacterial infection like mycoplasma and fungal infections that creates problem. In order to eliminate these, we provide a proper aseptic condition. Normally the cultures are maintained at the laminar flow chamber in a unidirectional flow in which the HEPA filter eliminates these microorganisms from them and maintains a sterile environment and make it contamination free. Further, the subcultures are taken up and they are collected and they adhere to the available substrate to reach the confluence. After a few days, the cells forms a monolayer and overgrow and gets crowded. So when it overcrowded, it is unfavorable for the growth and development because the bottom layer will not get sufficient amount of nutrients as well as a growth factor which is necessary for cell development. Further, the infusion of oxygen to the bottom layer will be less when as well as carbon dioxide which is maintained by the carbon CO2 incubator for the proper pH. So this is a problem. So how to identify whether the cells are present in the normal condition or in the live conditions or not, which is proper or not. So there are the cells are then removed. At this point the cells has to be removed by the subculturing process or passaging in which trypsin is utilized to detach the cells from the old medium. So when these cells are transferred to the fresh flask after centrifugation, the fresh media is added and again the splitting of the cells takes place for the culture purpose. The passage number refers to the number of times the cell line has been subcultured. So passage time is very important question. Passage time means how many times we take up the subculture and how many times we are transferred to the fresh medium, that is called as passage. Normally, in order to detach the cells from the culture vessel, we make use of the trypsin as well as collagen combination or by mechanical method by using scraper or shaking or vigorous pipetting can be involved in the disaggregation of the individual cells in the fresh medium. Once the cell line is established, the karyotype of the cell has to be determined and the karyotypes are vary from normal. Majority of the cells the culture have normal karyotypes are called aneuploid When the normal karyotype is desirable, the presence of the abnormal karyotypes has to be eliminated which are used in the in vitro studies. The normal karyotypic cells are used to create transgenic animals, and distinct care must be taken in handling those cells to avoid the minimal and avoid to minimize the chromosomal changes. And how to maintain these cells in a preservative condition. So once the cells are subcultured or primary cell culture, it has to be maintained in a very low temperature of minus 180 to 196 degrees centigrade. That is done with the help of liquid nitrogen. The liquid nitrogen is used to freeze the cells at very low temperature to form ice crystals, and it is reduced when it is reduced to minus 130 degrees centigrade. The freezing of the cells can cause mortality due to the damage caused by the formation of ice crystals. So we have to thaw the cells further in order to prevent the ice crystallization of the cells under liquid nitrogen at very low temperature. We are adding additional compound. We call it as dimethyl sulfoxide or